Hello, my name is Snehal Sharad Kadam. I am from MSc First Year Biotechnology and my topic is Structure and Properties of Nucleic Acid Bases. So, what are nucleic acids? Nucleic acids are long chain polymers of nucleotides present in nuclei of cell, so they are also called polynucleotide. They play an important role in transmission of hereditary characteristics and biosynthesis of proteins. They are the largest and the most complex organic molecules. Friedrich Mischer, a Swiss scientist in 1869, isolated an acidic compound from the nuclear material of salmon sperms and named it as nuclein, which is now called nucleic acid. What are nucleic acids? Nucleic acids are polymers, which is monomer of nucleotides. Nucleotides are made up of nitrogenous bases, sugar, and phosphate. These all combinedly form nucleosides. Nucleic acid made up of nucleotides. Nucleotide contain phosphoric acid and nucleosides. Nucleosides contain sugar, purine, and pyrimidine base. Sugar. The sugar in nucleotide and so in a nucleic acid is a pentose. In RNA and its nucleotide, the sugar is ribose, whereas in DNA and its nucleotide, it is deoxyribose. The prefix deoxy means without oxygen. Nucleotides can be hydrolyzed to yield nucleosides and phosphoric acid. This is the structure of sugar. It is ribose sugar and this is the deoxyribose sugar. There are two kinds of bases. First is pyrimidines, which are single ring bases. Second is purines, which are double ring bases. Purines contain adenine, guanine, and pyrimidines contain thymine, cytosine, and uracil. Hydrogen bonds. The bases attract each other because of hydrogen bonds. Hydrogen bonds are weak, but there are millions and millions of them in a single molecule of DNA. The bonds between cytosine and guanine are shown here with dotted lines. Nucleoside. This is the structure of nucleoside. Nucleoside, a compound that consists of D-ribose or 2-deoxy-D-ribose monosaccharide covalently bonded to a nucleobase by beta N glycosidic bond. Covalent linkage forms between N19 of purines and N1 of pyrimidines to C1 anomeric carbon of ribose to deoxyribose. So this is the structure of pyrimidines and purines. Phosphate group. It is negatively charged at neutral pH. Typically attached to fifth position, nucleic acid contain one phosphate per nucleotide. Monomers. Nucleotides are made up of three parts: phosphate, phosphoric acid, nitrogenous base, and sugar, ribose or deoxyribose. Nucleotide monomers are joined by 3,5-phosphodiester linkages to form nucleic acid polymers, that is polynucleotide. There are two types of nucleic acids, which are deoxyribonucleic acid DNA and ribonucleic acid, that is RNA. RNA is a polymer of ribonucleotides linked together by 3,5-phosphodiester linkage. Types of RNA. In all prokaryotic and eukaryotic organisms, three main classes of RNA molecules exist. First is messenger RNA, that is mRNA, transfer RNA, that is tRNA, and third is ribosomal RNA, that is rRNA. The other are small nuclear RNA, snRNA, microRNA, miRNA, small interfering RNA, siRNA, and heterogeneous nuclear RNA, hnRNA. DNA structure. DNA consists of two molecules that are arranged into a ladder-like structure called a double helix. A molecule of DNA is made up of millions of tiny subunits called nucleotides. DNA structure. DNA has four structures, that is primary structure, secondary structure, tertiary structure and quaternary. So we will see it in brief. Primary structure. The order of bases on the polynucleotide sequence. The order of bases specifies the genetic code. Secondary structure. The three-dimensional conformation of the polynucleotide backbone. It is also called as double helix structure. Tertiary structure is supercoiling. Quaternary structure is interaction between DNA and protein. Difference between DNA and RNA. In RNA, it is a single stranded mainly, except when self-complementary sequences are there. It forms a double stranded structure, that is hairpin structure. In DNA, there is double stranded except at certain viral DNA which are single stranded. In RNA, ribose is the main sugar, and in DNA, the sugar moiety is deoxyribose. In RNA, pyrimidine components differ. Thymine is never found except in tRNA. In DNA, thymine is always there but uracil is never found. In RNA, being single-stranded structure, it does not follow Chargaff's rule. In DNA, it does follow Chargaff's rule. The total purine content in a double-stranded DNA is always equal to pyrimidine content. Properties of nucleic acids. Nucleic acids are insoluble in alcohol, slightly soluble in cold water, but readily dissolved in hot water and dilute alkalis, forming alkali salts. They are precipitated by HCl and by excess of acetic acid. Fusion test differentiates the DNA from RNA. If the deoxyribose sugar is present, a red color is produced with the dye. Ribose sugar do not exhibit this reaction. Hydrolysis of nucleic acid gives nucleotide, which can be considered the unit that make up the polymer. Biological functions of nucleic acid. DNA is the chemical basis of heredity and may be regarded as the reservoir of genetic information. The proteins are synthesized by various RNA molecules in the cell, but the message for the synthesis of a particular protein is present in DNA. DNA is exclusively responsible for maintaining the identity of different species of organisms over millions of years. Thank you.